أسعد الله مساءكم لكل خير بكل خير. Welcome everyone to this Institute event. My name is Maurice Pomerantz. I am the Executive Director of the NYU Abu Dhabi Institute, which is a center for scholarly exchange, academic research, and public outreach. Tonight, I have it's my very great pleasure to only have one obligation, and it's a very pleasurable one, to introduce my colleague, Eliseo Ferrante, who's a visiting professor here. He received his Master of Science from Politecnico di Milano and one from the University of Illinois at Chicago in 2007, both in computer science and artificial intelligence. He received his PhD degree in applied sciences from the Université Libre of Brussels in 2013 and a thesis in swarm robotics and statistical physics, which sounds like a very interesting topic. I hope to hear more about swarm robotics one day from him. But today, we're going to be hearing about other things, the Internet of Bio-NanoThings, and I'll leave it to Professor Ferrante to introduce the speakers. So welcome everyone, and I look forward to seeing you at future NYU Abu Dhabi Institute events. So thank you for coming.
Thank you, Maurice, for the very nice introduction, and good evening, everyone. My name is Eliseo, and it's an extreme pleasure today to welcome Professor Ian Akyildiz to NYU Abu Dhabi. Professor Akyildiz got his education in Erlangen, Germany, but then most of his career is spent in Georgia Tech, where he has been tenured professor for over 35 years. Is one of the world's most known experts in wireless communication and has been very active around the world and held in position in different countries, including Spain, Finland, Saudi Arabia, and recently Ireland. He is also advisor at Technology Innovation Institute in Abu Dhabi, and that's why we have a pleasure to host him very often in the UAE. One thing I wanted to share about Professor Akyildiz personally is one thing that impressed me is the fact that he's able to he has a very strong legacy around the world. He was being able to build a network of networks. What I mean is that there are many well-known professors around the world in his field that have been once the Professor Akyildiz student. I can personally experience how he can achieve this. He's one of the best mentors I ever met in my life. He's like really wishing the best for his students, for his collaborators, pushes hard for you to achieve your accomplishment. So, so please help me welcome Professor Akyildiz to the stage.
Good evening, and first of all, thanks to Eliseo and also to NYU Abu Dhabi for hosting me. And they asked me what topic I prefer, and then I suggest some topics, and they picked this one because it sounded more futuristic and more interesting, not the mainstream presentation. So the title is, you saw the longer version of it, but the shorter and more technical version is Internet of Bio-NanoThings, and which the terminology I coined back in 2008 with my PhD students. And we did a lot of work the last almost 20 years. And the last stage of this research was or is how to detect, avoid infectious diseases. And that's what I will present you today. And so let me give you a brief background story. Back in 2006, when I was producing 4 PhD students that time, they were on the research market for professor positions, and they all came back and were complaining that all departments in the US were looking for nanotechnology people. And then I got interested to know about the nanotechnology, and I started to study it. And because I remembered my time back in early 70s, the microchip era, and how much it impacted our lives. So I got interested and I started to work on it. And that time, Fernando Brunetti, the second person, and from Madrid, and Cristina Blasquez from Barcelona, the poor lady, young girl, she died recently. And they came to my lab as visiting researchers, and I suggest them to work on these nanotechnologies. And I'm a communications professor, telecommunications. And my interest was more from the communications perspective. And I realized that in the nanotechnology, majority of the work was done on the nanomaterials and also on the nanocomponents of computers. So my interest was more based on communications, and I wanted to know how these devices will communicate when everything is out on the market. So that's why this interest came. And then I realized that there is a perfect communications paradigm in the nature about nanoscale machines, which I will present to you today. And then we realized that also we can, it's kind of like biology-inspired nanomachines. And then also we said we can also create nanomaterial-based, like graphene or metamaterial-based nanoscale machines. And we look at their communications problems. And then those years, in 2007, I hired Massimiliano Pirabon, the second guy, and also his name is there too, somewhere, Josep Jornet, so from Barcelona. And then I started the research group. And then in the meantime, received many projects all around the world. I started a research center in Barcelona, even in Saudi Arabia and Jeddah. So anyhow, so I will present to you. So we also work with some other disciplines like biomedicine and electrical engineering, computer science, physics, etc. So all their names there. So so this is the kind of like a list of references. So I mentioned to you, this is the work that bio-nanothings, as you see here, the human body has all these nanoscale machines inside the body, like neurons, hormones, and calcium signals, and bacteria, molecular motors. And when you are interested in these Nobel Prize, I always say Oscar, Nobel Prize, majority of the people in the medicine are receiving Nobel Prizes within these concepts. So you can check that also. And so these nanoscale machines in the human body are communicating in the human body. And so then, you know, how can we get the information from the inside the body outside, number one. Number two, can we also create nanoscale machines like as neurons, like artificial neurons, artificial calcium signals, artificial bacteria, and then also solve many health problems. So that was our objective. And then also get the information out what's going on through some gateways which will be on the, like outside the body, and then information will come in and we'll use the internet, go back to the physician's offices. So this entire concept is a closed loop concept. It's called Internet of Bio-NanoThings. So we published you know, one of the first papers in 2015, and then we continue. So I mentioned to you there are two directions. One is the nanomaterial-inspired nanoscale machines. You see there that like a perfect computer, like all these components like processor, antennas, memories, and then you see also these brushes. They're zinc oxide nanoscale batteries, but there are also now new battery technologies that they can create this very you know, tiny nanoscale machine. And for the material, the graphenes, which is invented by Gaim and Novoselov, two St. Petersburg Russian people. They got Nobel Prize in 2010. And then the other way is the bio-inspired, meaning can we create bio-inspired, meaning artificial molecules or artificial machines. So again, you will see what I'm talking about when I talk about bio-inspired. So we will be focusing more on the bio-inspired concepts here. So let's look at from the nature perspective. So there are these so-called eukaryotic cells, like human cells, and prokaryotic cells. A typical example is E. coli bacteria. And these are perfect nanoscale, precise nanomachines. And also, they can communicate, interact, and cooperate with each other. Like you see here on the left-hand side, eukaryotic cell tissue, and on the right-hand side, you see bacteria population. And so they are really communicating with each other. There are like billions of them, and then they communicate with each other, and they create like swarm of, cells, or a swarm of, or it's also called in biology quorum sensing for the bacteria population. So let me show you the, inside of these eukaryotic cells. Like you see in the middle, there's so-called nucleus, and there are also ribosomes. They are perfect example for biological memory for the ribosomes, and the nucleus is a processor. And then there are these mitochondria. They are biological batteries, like energy sources for the cells. And there are chemical receptors, there are biological sensors, and molecular receptors, because they are communicating through molecules. And then they're also called gap junctions, and they're a perfect example for transmitters of molecules. So there are, as I told you, millions of them, and they communicate with each other. So let's look at the prokaryotic cell, like bacteria. If you don't like biology, I'm really sorry. Anyhow, so the prokaryotic cells are a little bit mishmashed, not clear like the human cells, eukaryotic cells. They still have these, the nucleus and plasmid, like here you see in the middle, and they're like perfect example for nanomembrane processors. They have also so-called pilies, like they are like sensing, nanosensing units, and also actuating units. There is also cell membranes, which is acting like nanopower unit, like battery, and protein receptors, like nano receivers. Ribosomes, they also have as acting like transmitters. And there are these flagellums, like tails. They can navigate them. So this is a perfect example for a bacteria, or especially E. coli bacteria. But I have to say that, they are moving very, very slow. So when you look at them under this platform, like they are very, very small, and then when you feed them sugar, they populate. And then when they don't get sugar, they die. Okay, so it's very interesting. So again, just to give some more examples for the mitochondria, which is the biological energy source, battery. So what they do is these mitochondria obtain glucose, amino acids, fatty acids, and oxygen, and they synthesize so-called ATP, or adenosine triphosphate. So just let me give you an example. When you're tired, so you say, I need a free, I need air, right? You go out, you get oxygen. So you charge your mitochondria. Or the doctors tell you, don't feed sugar or chocolate to your kids just before going to bed, because their cells are getting charged and they get a lot of energy, right? So that's a typical example how these cells are charged with these mitochondria. So here is the biological memory and processor. The DNA is in the nucleus, contains the information of a protein structure, like it acts like memory. Since you are in the computer science, you know the Turing machine, one-band Turing machine. That's this DNA strain. It's like you can read all these DNA values. And then there are these so-called ribosomes, as you see in the middle, like the Turing machine read and write things, right? It reads and processes the DNA information and acts like a processor, and they synthesize the proteins. And the proteins control the functionalities of each cell, and for example, cell signaling as transmitter and ligand binding as receptors. So all I'm trying to show you is that these cells, right, as alone, but then they communicate with each other. Like in the eukaryotic cells, as I mentioned to you, we have so-called molecules. Like, what are the molecules? Like proteins, ions, and hormones, depending on the nature of the cells. And then they get packed with the messages. We'll talk about those. And then they flow from one end to one cell to another cell. I mean, it looks like long distance, right? It's not. They're all close to each other, right? So then prokaryotic cells, like typical example are the bacteria, like the transmitters and receivers are so-called molecules. As an example, auto-inducer exchange for quorum sensing, meaning these swarm of the bacteria. So now there are these some names in the biology. So now the bacteria comes, and then the transmitter, the cell, puts into the information into this plasmid. It's like the intermemory, it writes there. And then these bacteria are slowly moving, tumbling around, and then they can also, this is called chemotaxis process. And when they reach the destination prokaryotic cell, they mate. Then it eliminates. Suddenly, you can see that, you know, even with naked eyes, even they eliminate. That means it can put the information sent to that bacteria into the plasmid of that bacteria. So that process is called conjugation. Okay. So as I mentioned to you about also these transmitters, like these cells, for example, release these molecules, right? And so they float, and like as I told you, the gap junction was acting like transmitter. They float, kind of like diffuse, and then they move towards to the destination. And the destination can try to suck those molecules. When they are successful, that's called ligand receptor binding. That means the molecules with the information are received correctly. And so these are kind of like chemical receptors, right? So this is a perfect example for how a transmitter and receiver works inside of the human body, for example. So what we try to do is we try to create this so-called molecular communication theory. You have seen that, you know, transmitter, receiver, like wireless communications, and then the channel model here, right? And source creates these molecules. It goes through the channel, and then it's received by the destination. And we try to find out what is the effect that these molecules with the message loaded are received at the destination, what type of effects they will have at the destination. So let me go to these, you know, the way that the communication theory people may know about electromagnetic communication and acoustic communication. They are usually waves, right? Sine waves and etc. And then also in optical communication and quantum communication, we also have these waves, as you see here. But in the molecular communication, we are really so-called particles, and they diffuse in the air. They just go to, you know, sometimes they get lost, they don't go to the destination, and some of them are sucked as the ligand binding receptor by the receiver. Okay. Now, after this brief introduction into the biology, the question here is, can we create man-made, man meaning, you know, human-made biological nanomachines? The answer is yes. That means we can reprogram these artificial cells and via DNA manipulations, and that's called genetic engineering. And that will really help us a lot. In fact, unfortunately, some maniacs in the US found all my work the last 18 years. I'm getting a lot of hatred mail, so my life is in danger. They think that I'm Dr. No in James Bond movies, that I'm trying to manipulate human lives, but it's, you know, I'm not the only one. There are many people working on this subject, you know. So, so now what we try to do is, so you have seen the bacteria, right? So what we can do is we can try to genetically engineer this bacteria, and you know, there are many illnesses, as you know. I will point them out to you, you know, based on bacteria or viruses, right? So if I create genetically engineered healthy bacteria, for example, Crohn's disease, you know, I don't know if you know that, it's an awful disease for intestines. So there's no cure. So I can inject into the body, and then they can fight these ill cells or, you know, ill bacteria, and then solve the problem, you know. So just an example. So I hope you are getting the point, right? So we are trying to create these nanoscale machines, genetically engineer them. They will act like bacteria, and also in the future we will see that neurons and calcium ions and etc. And it will help us for prolonging our lives. In fact, the mRNA, the COVID injection is exactly based on this idea. So all these fluids, right, there are nanoscale machines inside, right? So when you look inside. So long-term goal is, so this like doing the prokaryotic cells, like bacteria cells, are easier to manipulate, genetically engineer them, but the human cells are much more difficult. Of course, some people may say it's also ethical or unethical, but that's not the point now. So on the long term, we try to create genetically engineered human cells. And, you know, the success is already there, some of the cases. So how we do this here is we insert a piece of a DNA code, which is called lentil virus cell, and which is a special type of virus used in genetic engineering, as you see here. And then this lentil virus is able to infect the eukaryotic cell and inject a new DNA code in the cell DNA. And then this cell is a healthy cell, and it can go and fight the ill cells, and hopefully will solve the any illness problem, whatever we see here, right? So that means all the cells will be replicated and solve all our problems. So now I recommend to some of the young people, the humans are really fantastic to see. You think that you got the idea, but then you find out that somebody did it 100 years ago. So there is this guy, Paul Ehrlich. He's a Nobel Prize winner in 1908. So he had this idea before, right? Really. So he called it, he was from Austria, Germany. He called it magische Kugeln. That's German meaning magic bullets. Exactly this concept, right? So these are ideal therapeutic agents, he called it that time. And then he injects into the body, and then he targets diseases, and then solves the problem, right? And then many of these, later medicines were using this concept, but not fully, right? So that's why now we are in the time that we can use these ideas fully. So this is the picture. The idea was I started a research center in Finland, in Tampere, and I got a big, large project about Finnish Distinguished Professor program from Finnish Academy of Sciences. And so when I started this center, there's a newspaper, Aamulehti. So the guy came and he had like 6 pages long interview with me. And then he put me at the wall, and then he put this picture. Everything was in Finnish, of course. So the point of this picture is, of this slide, is any illnesses you have, or we have nowadays, right? Alzheimer's, epilepsy, depressions, we can solve them. And then, because we can create these healthy neurons and then manipulate the, in the past they were electrocuting the poor people, right? So it's crazy. But anyhow, but now they are pumping in all the medicines, and they're like zombies, unfortunately. And then you can forget the diabetes problem, and then heart problems, and then cancers, for example, and diabetes I mentioned to you. So anything that you can imagine, hopefully we can solve these problems. And once the entire technology is out. So this is another example that we did for Samsung, believe it or not. Samsung is you never believe they were interested in this, right? So what we tried to do is here is same concept that normally what happens is either we take the medicine tablets or we get injections, right? For any problems. Like for example, I have this inherited high, what is it called, cholesterol from my mom. So I was taking tablets, but then it caused some muscle aches. Now there's an injection, they give it to you, and it's damn expensive. But so what you do is, the problem is the following. So now you have a target location, let's say, for any type of illness. And then when you take the medicine, like here, for example, so they go through the body, right? So the body is like a information highway, you can think about, right? So the medicine moves around your organs, and they also go through these veins, for example. They lose their effect because as they move, most of the time when they hit the target, already 40–50% are the effectiveness is gone. So now what we try to do is here, using the same concept, saying that, like from the system modeling theory perspective, normally you have input, box, operation, and then output, right? So then you try to do objective function output, try to optimize it. So we try to do the other way around. So we know the output, the delivery of the drug, how effective, how much dosage it should have, and then analyze backwards, like how much is getting lost inside the body, and then we try to find out how much dosage we should inject into the body of a patient. And of course, this is also going very well with the future medical treatment. Everything will be personally tailored to us. It's all is still very primitive level. That means you go to doctor, I have high blood pressure, or I have cholesterol, they prescribe you this, and then sometimes you have side effects, sometimes it's not helping you, and then try this, try that. So what we do is we will try to tailor all the medicine based on you, all your DNA and all your, all the activities in your body, and then we'll prescribe you or tailor you the medicine. So that's really the future of the medical field. And I don't know that we have medical people here, but I hope they agree with me. So we had this, one of the big projects was from the National Science Foundation. As you see here, we had, I collected many professors from different departments, from biology, from medicine, from computer science. So we were like 5 professors in this case, or 6. And we had also many PhD students and postdocs. And then we did this work about how the bacteria will communicate. We tried to understand the communication of these bacteria. Some of you will maybe think that, why do you need all this, right? So when you go to biologists and biomedicine people, majority of them are working with trial and apply. Like there is always something experimenting, measuring, try this, try that. Instead, developing some mathematical models, simulation models, kind of like communication models, and understanding that it works, and then you can manipulate this model, and then you can help them instead of always they keep creating another experiment. That was one of the major motivations of this work. So we did all these communication models, but then I also had these biology and medicine people. They set up the physical experiments, and then we try to match all the theoretical models with the measurements of the bacterial communication. And of course, in the theoretical models, you can do anything, right? But in the practical, you have to do certain things to match the theory or vice versa, right? But it worked, and so it was, you know, one of the very successful NSF projects that NSF people presented to the parliament, yeah. And this was like 2016, like 5-6 years. And I also used, you know, these ideas in other countries, as I told you, like Finland and even in Saudi Arabia, in Jeddah. We got money from CACST, Saudi Arabian Foundation, and our Barcelona, I started this center there. It's very successful now, Entricat. So we were asked with my people. One of them was my postdoc in Finland. The other one was my PhD student. He's professor now. Those 4 guys, in the meantime, as Eliseo mentioned, they are super successful professors. They were looking for jobs, right? So they are professors in the USA. One of them is running the POWER program, Platform for Advanced Wireless Networks Research, like hundreds of millions of dollars, an Italian guy, Tommaso Melodia. And then, you know, they're all successful now, so they're all full professors. So, you know, we were invited by these proceedings of the IEEE journal saying, what's going on now in this field? The reason is what happened is, 2018, 20 years ago, when I started this, all this theory was open. So we did all this research, and then many people came later, like 2015, 16. They kept writing papers. That's the problem in our field, really. So they keep writing epsilon, epsilon, epsilon. So we are dwindling around, right? So now these editor-in-chief asked, you know, can you somehow tell the people what to do now? So that's why from theory to human health applications. So we said that all this work is, you know, trying to analyze the diffusion process of the molecules. We did molecular motors research, we meaning all the entire research community, right? And then gap junctions, study calcium ions, chemotaxis, etc. And then from the theory perspective, again, in communications, you know, the capacity of the channel, or what type of modulation coding techniques, or also communication protocols. Many, many papers have been written, so it became a paper writing race. Now, many people are still writing papers, right? So it's very sad. So, because it's time now to do practical work, right? But it's more difficult. So I did work with my people, calcium signaling. You see all these publications, right? And then also molecular motors and fluid dynamics. That means what we try to do is, I told you about these molecules floating around, right? But normally what they do is, there are these so-called microfluidic tubes. You can see here. That is also a perfect example for veins in the human body, right? So then we try to analyze these bacteria movement in these microfluidic fluids with my colleagues in the mechanical engineering department, and try to find out what's going on. Like, you know, what happens is, so there are chambers, so you feed the bacteria with sugar, glucose, and they populate, and then they get out of the chamber. I hope we have some biology people that understand. So then they move in this channel, right, in this area here. They go to the next chamber. So it's kind of, this chamber becomes the transmitter, the other chamber becomes the receiver, and then we try to find out how many survived and reached the destination, for example. So that's, we are trying to find the analogy between communications and the reality here. So what did we learn, you know, from all the studies? So what I'm trying to show you here is, although I show my work, but you know, there were hundreds of other papers, right, as I told you. So what we did in this paper that we were invited to write is to look back and tell to the community what are the lessons learned, right? So the practical and useful applications of the molecular communication theory is still very limited, and we need novel perspectives and novel ideas for actual applications of these models, right? So, so we should apply the information communication technologies for human health, right? And somehow we will be concerned with acquisition, storage, analysis, modeling, and exchange of patient health information. And we need to look at through some silico computing platforms and also the internet, right? And so this is really interesting because this theory that we were trying to establish, it can enable the sensing and control of this information from or inside of the body, because it propagates in vivo through the biochemistry of the patient's bodies, its cells, and their molecular content, right? So now, here's an example. Unfortunately, many people have this cancer problem, right? What happens is they arise from abnormalities in the propagation of the molecular information underlying cell differentiation and proliferation. That's really that causes the cancer, the cells become cancerous, right? So again, without boring you again, too many details. So we try to come up with this, the system model, the theory, and the application case. The theory is that we will have these theory and tools already established. We apply the model at different scales, the information flow in the body, as I mentioned to you about all these molecular motors, neurons, etc., bacterial communication, and down to tissues and single cells. And then we move to the so-called molecular communication engineering. And that's what I was trying to tell you, trying to create synthetic cells. It's called synthetic biology. So now we go like this way, and we are creating these synthetic cells, and we redefine, correct, and control the information flow through these synthetic or artificial cells to enable novel interfaces to the internet, meaning to enable the information getting out of the body and sending to the internet. Why? You will see that. So that we'll get some cure from the physician's office or whatever. You will see that. I want to show you that, okay? So there are these in the natural systems on the left side. We have body system scale, right? Organs and tissues are interconnected to perform a certain function. Then you go down to the cellular scale, where cells coordinate with each other and process information. We go one more down, that's called molecular scale, where information is encoded into substances and exchanged through chemical reactions. As I showed you in the beginning also, like, you know, all this chemical reaction, right? The receivers reacting them, right? Ligand binding. So on the other hand, on the right-hand side, we go from up, down, up, bottom up. So molecular components, communication components here, they genetically program tools from synthetic biology. And then we have molecular communication devices. They're engineered microbes or human cells capable of pre-programmed behavior. And then at the end, we have a network of these molecular machines, and they communicate with each other. And you know, these are all, again, so-called synthetic or artificial cells. Do not forget that, please. Okay? So the message what we are trying to give is that we need further research in this theory. We should couple them with experimental validation, especially for future technologies that will be subject to clinical trials. So there are many challenges, right? That's what we need, really interdisciplinary research teams, incomplete biological knowledge, individuality and variations, because each person is different, as I told you about tailoring the medicines per person. There are mutations, evolutions, safety and security, and also ethical concerns. That's why those people are sending me hate messages because of the ethical issues. They think that we are manipulating human lives. So we will need some innovative solutions for health. And there are many grand challenges. For example, that's really the 21st century grand challenges for engineering and medicine, engineering better medicines, and advancing health informatics as defined by NAE, National Academy of Engineering. So, now coming to this topic now with all this, I try to shortened everything in the last 18 years, about how we can use this theory for for example, early detection in infections. Okay, so this is, you see a bunch of people here again. We had also people from Emory University, which is a fantastic medical school at Georgia Tech. We had also Johns Hopkins. They're also top in medical field. So I created this large group of people. We also receive funding in the USA. So the problem here is early detection of infections is very critical for patients. Like cancer patients, the chemo patients are very vulnerable to serious bacterial infections. Okay? And also cystic fibrosis. Infections come wave by wave, especially for young children. And they die. And there are many people, they die from sepsis. That's a typical bacteria disease that kills the people, right? So the project, I called it Panacea. I take the credit for it. Panacea is the remedy for everything. It's Latin, right? Remedy for everything. So continuous monitoring of bacterial infections with this IoBNT, Internet of Bio-NanoThings concept. That's what I will present to you next. So what we are trying to do is we want to early detect any infections and then timely administer the antibiotics, and also tracking the efficiency of the antibiotics. Why? Reducing the mortality and also the hospital costs. The hospitals are very expensive in America. It's unbelievable. So I get like echo measurement for my heart. Before I take long trips, I travel all the time. So I go to check my heart. I don't want to die in a hotel in the world. So, you know, that simple thing, like 20 minutes, they measure the things, $10,000. So my share is $1,000. So I still pay $1,000. So it's very expensive. You know? Yeah. Welcome to America. So the first application is infectious in immunocompromised patients. So it's the same thing, right? It's called Pseudomonas aeruginosa. Infections are very problematic, like sepsis, hospital-acquired infections, cystic fibrosis in burn patients, chemo patients, and unfortunately, these cases are very resistant to a large spectrum of antibiotics. And also, they can infect various organs, such as lungs, urinary tract, kidney, skin. That's why you die, you know, unfortunately. So don't say, bacteria, I can just take some antibiotics, and it's not that easy. I have many other applications. Another one is for the military, for the US Army we did this. Soft tissue infections in military personnel. It's called Acinetobacter baumannii infections. And same concept, right? And so what we are trying to say is, that's why I called remedy for everything. Panacea will solve all these problems when we realize everything. So here's the big concept, Panacea Architecture, right? So we talked about before all this inside the body, right? So now what we're trying to do is we create this bio-nano thing. Also, I take the credit for it. Remor means in Latin voyager. So it's a special designed nano device, or, you know, actually it's a machine, right? So it can be either biologically inspired or nanomaterial based. So I mentioned to you in the beginning, right? So we call these remors, and they will be in the body, okay? They will collect information, right? Like, for example, any infectious diseases or anything. And then when they find any infectious diseases, they will get that information. They will use a gateway, like tattoo type thing, which I don't like tattoos, but anyhow. So it's like very like Band-Aid type thing on the human body, you know, on the arm or anywhere. And then it will collect the information from the chemical world, right? This is the chemical world. And then it will transmit that through the backbone internet. That's why we call it cyberspace, and like mobile device. But then this area is electromagnetic communication, right? We are this typical communication field. So this gateway must do some reprocessing, right? From the chemical world, you will take all these and then convert them into electromagnetic waves, or vice versa. Electromagnetic waves will come, it will be converted into chemical waves, chemical particles, and then will be transmitted to the area that we need to address. So now the cyberspace, like there are these emergency actions, right? If there is some emergency, the CDC is the Control Disease Center in Atlanta. They're based in Atlanta. They were very famous during the COVID time. And then clinical database, or clinic, and also like physician's office, right? So now what we are trying to do is a closed loop. So when like each of us can transmit our body, intrinsically, information to your doctor, right? Your doctor can get alarmed if there are some abnormalities or anything alarming, and then doctor can take a look at it, and then see, and then also all the information collected before, they are stored in a process stored. And then we will use AI machine learning techniques. We'll download that information from the past about the patient and the new information. The doctor can decide what to do. It can either retransmit signals to the human body and tell to the remorse, do this action, that action. It sounds very science fiction, right? But it's really true. And then, or if it cannot do that, so it will, definitely, go to the patient or call the patient and then do, in-person treatments, right? So that's the big picture of the Panacea architecture. So the project had these work packages, typical. I just want to produce, show you the big picture here. So on the left-hand side is all theory. On the right-hand side is all experiments with the medical and bio people. So the left-hand side, what we do is the following. First of all, we design these remors. Design and produce, right? Design of bio-nanosensors. That's what we will inject inside the body, right, this year. And then we need to some kind of find sensor interface chip of remor, meaning the information collected, remor must send it through the body, like that gateway to outside, right? That's the interface chip. And of course, the remors will also communicate with each other. Don't forget that they will create a swarm of these remors, bio-nanosensors. And then how will you integrate them, meaning how will they communicate? All these questions are the work package 1. And work package 2 is that how do we model infections and drug delivery? You have seen the Samsung case I told you. We use that model or those models for the drug delivery part. Remember we said if we find out that there's some irregularity or some problems, we will also do drug delivery, right? So that goes with that task 2.1. And then another question here is that we call this near field wireless data and power transfer, meaning like from the gateway, you go to your cell phone, right? It's a very close distance. And they will call, it's called near field, like very close communication. And they will be doing, communication in the electromagnetic part. And then in this part, like interface chip, will do this conversion between chemical to the electrical world. And then another task here is all the backbone, like internet communication problems. Okay, and then the third package is totally based on data processing, right? So how do we collect all the data? There are billions of people, right? So all the information will be collected for each person. And then many of the information may be similar to it. Maybe we can aggregate them, fuse them, in order to reduce the volume of the traffic or collected data. And also, like, we will store them, right? And also we need to access them fast. So another question here is, how do we do early detection of these diseases or infections. As I told you that we have all the collected information in the storage, and then we may use deep learning, machine learning algorithms to learn and then apply those to detect these infections. And then learning-driven drug delivery and data visualization. And then last one is, of course, secure and privacy-preserving data handling and transmission. The right-hand side is testing of individual components of these remors, then prototyping of Panacea in a vitro test bed, and then the third one is integration of all this entire Panacea in a vivo test bed. Vitro and vivo, I hope you understand, right? So, how long time do I have? 10 minutes, 5 minutes, or what? How much? Okay, so I should, yeah. I'm afraid I hope I'm not boring you here. It may be too futuristic. So what we do is, so these remors, nanosensors that we design and or fabricate, genetically engineer, they measured quorum sensing, because we're talking about bacteria, right? Quorum sensing communication representing infections, like here is the infected area. So one solution is to develop everything electrochemical sensor, okay? And there are functionalized electrodes directly bind with quorum sensing molecules, like here you can see, and then collect that information, and then we'll transmit these little things, right? That's all like here on the right-hand side. B is the bacterial sensor case. So this is the typical that these molecules are transmitted. They will go to the interface. So another solution, we try again different ways of remors. Another solution is called bacterial sensor 1. That is, this is kind of like, you can think about, this is the nanomaterial based, like material based, and these two tracks are natural base or bio-inspired base. So like engineered bacteria here, right? And this, we call this engineered reported bacteria, produces an electroactive product upon reception of the quorum sensing molecules from here. And then they generate currents corresponding to the concentration of the quorum sensing received. And this one is much more difficult, again, based on the biological inspired bacterial sensor. Again, this bacteria reporter produces bioluminescence or fluorescence upon reception of quorum sensing molecules. Remember, they eliminate, right? And then what happens when they eliminate, they're binding. And then we try to find out through a light sensor, generating current corresponding to the concentration of the quorum sensing received. And then we need to maintain live bacteria inside the sensor, which is very difficult, and the sensing process is noisy. That's why we have problems to perfectly apply these solutions for the time being. So what we are trying to do is, again, in a nutshell, maybe it's too much detail for you, that these bacteria-based or material-based sensors will try to always sense the bacteria in the body. If they have some irregularities, it will collect them, detect them, and then send the information out that there is a problem of the bacteria infection. That's the explanation of it in a nutshell. So then we have this chips, the interface, as I told you, like all these sensors I talked about, remors, will transmit to the, to the gateway on the body, and then from the gateway, we'll use, some watch type or, tattoo type thing to your cell phone. That's why we call it near field. You can see the skin here, right? And then we'll use through the cell phones, etc., go to the cyberspace, to your doctors or to the CDC, etc. So again, you see this closed loop here. And then, so this design has, again, maybe too much detail for you. We're using ASIC technology. And we have, of course, ultra-low power design as well as ultra-low noise, because the biosensor outputs are very low voltage and also the currents. So that's why we have this design also. And then we are using different communication paradigms inside the body with chemical, then from the body to skin, magnetic induction, also from the skin to the cell phone, and then rest is the electromagnetic communication. So I show you all of this. So the last modeling is, again, we use all the communication theory that we did before, and like MIMO, molecular communication channels, like many antennas, because they're communicating, and we capture all the time-varying channels, and shadowing by the tissue structures. That's typical communication problem in the wireless communication, shadowing and multipath. We also capture all of them. So you can see here why I call this MIMO. So one colony here with bacteria, right? So it's like one antenna, for example, another one here. So multiple of these, or even, you can do distributed MIMO, or one site has multiple antennas, right? These red dots. And then they will, be sensed by the remors, collected, like that's why we call this MIMO communication. So then, we did all this work. And so then the third one is the cyberspace of IoBNT. How do we effectively collect data and deliver to the cloud? Not one question. Then, as I told you that each person is different, how do we capture this bacterial concentration, which reflect the infections for each person differently? How do we detect infection from the data quickly with high accuracy? And also patient health data is very sensitive. That means how do we convey all the data to the physicians or clinic securely and safely, right? So that privacy will not be affected. So then, next question is, I told you there are millions of, billions of data, and how do we collect them? Then can we aggregate them because they are similar to each other, and we need to come up with visualization algorithms so that we can see what's going on. We use some cutting-edge deep learning techniques, and of course security and privacy is again very important here. And then the aggregation, we told you about this again, about deep learning, learning-driven drug delivery and data visualization. So we have these two types of drug delivery. One is called active drug delivery, which is automated antibiotic and enzyme delivery by implanted or dressing devices. Another one is called passive drug delivery, which is alerted patient, which has these infections or possible infections, takes antibiotics orally by the suggestion of the Panacea system or the doctor based on the information collected from the Panacea system, okay? So in other words, we try to enable human-machine interactions between patients, caregivers, and this Panacea system that we are trying to create, okay? And so then, as I told you about early infection detections, I'm going backwards. It's getting tired now. So security and privacy, as I told you, is very, very important in this field, in the medicine especially. Can we do hardware-embedded security? And then authentication, confidentiality, integrity, and availability are important. Can we design some lightweight asymmetric authentication protocols? Then privacy-preserving data storage and processing protocols. We suggest using some homomorphic encryption techniques, and also trusted execution environments, right? And the testing, so we tested, you will laugh now about our testing environments. The babies are also starting small, right? And small steps. So the first step was, we kind of like learning and, you know, applying these ideas here. So the experimental was, so we had this remor, we put it in a tube, right? And we wanted to measure the quorum sensing molecular concentration in this tube. And then we injected some molecules into this and then try to catch, that remor catches these drops which is injected into this tube. And then the remor will transmit that information to the laptop here and then understand that, you know, what is the effect of this, for example, diseased drop of some bacteria cell, for example. So then another one was, that's also very funny, right? So same concept, but we put a meat in the middle that's just emulating the human body, right? So now you can see that same concept, and then the remorse is sending through the meat, trying to avoid the, you know, kind of like emulating the human body. And then we have a gateway hub, as I told you, exactly what we are trying to explaining you, doing this in the experiment. And then we, you know, with the near field communication, we send it to the laptop and understand how much is captured here. And the third case was that it's little, and if you like animals, I'm sorry, but that's life. So we have these little rats, and my colleague has a bunch of them. So we had, you know, we injected, no infection, and then we put remorse here, as you can see here, inside the rat or mouse, whatever you say. And then we had some lethal dose, and then we had lethal dose. And then we tried to capture through our remor that, you know, that this dose is, you know, that remor is capturing that infection, for example. And then again, we, you know, emulate all these things here and collect that in the destination. And then we try to make some, you know, what is it called? Closed loop here again. So that's where we are. And then we had this COVID problem, you know, and so we had some delays. Unfortunately, we couldn't be in the labs, and it messed up a lot of our projects. So we are trying to, you know, reestablish everything. It lost our 2 years of time. And also the team is spread out, you know, so I'm retired, you know, some people left, and so anyhow, so we are working. And then I used the same concept just before the COVID in Norway. We applied this project, we call this, it's a smaller, shorter version of it, and they applied, but it was more theory. Although it's like with the University of Oslo, Rikshospital, so we mostly try to do the theory applications, I mean theory here in Klippeås. It's also done by this year. So we did all through Zoom, and, you know, theory is easy to do when you're in the office, I mean in the home office. So now, when we were doing this, you know, this started back in 17, right, 2017. And then, so you will laugh now, 2016, I started this very large project for Norway, and I called it Coronal. I'm not kidding. And Coronal, and it comes from the cell, whatever, you know, short form, Coronal, but we knew that Corona existed. But we were laughing, you know, and so it was 50 million euro project with Rikshospital, Oslo, and then Trondheim, and so this Norwegian council gives these large center of excellencies. So we want to do this virus, you know, same concept as I told you before for bacteria, we want to do for virus. And so we worked like 2 years on it, and 17, we were one of the last finalists. So I had the interview, like third steps, and I went to interview, and there was a international panel, and so I had this colleague there. He was younger than me, and the panel asked me, "Will I come totally to Norway. And I was professor at Georgia Tech, and I didn't want to lie, right? So I said, no, he will do it here, but I will be coming and going. And that killed us. They said, I mean, they didn't say openly, but he said we found out that they said he's too inexperienced to run this 50 million euro project. So we didn't get it. But then what happened is karma, right? Virus hit. I wish we did, yeah. So, yeah, I'm sure they, anyhow, that's their problem. So anyhow, you can see bacteria and virus have different worlds, right? So like viruses are much smaller and they are non-living. So they bind somewhere and they suck like parasites, right? Viruses, and that's why it needs a living cell to reproduce themselves. Bacteria are not like that. Bacteria need only sugar, right? Glucose. We have glucose inside the body, right? So anyhow, viruses are not beneficial. Bacteria are beneficial, some of them, right? We have all bacteria actually. But then the viruses are not. So viruses are really awful, right? And then they can be prevented with vaccines. You cannot take antibiotics, right? And normally they give you steroids. It's awful. That has all these side effects, right? And then SARS was there 2003 in China, and then there was also the Africa. What is that called? I forgot that now. Ebola, right? Who said that? Ebola, yes, Ebola. But then we had this COVID, right, or corona. So that's why those people, they found all the things saying that, these guys knew that. In fact, 2 weeks ago, I came from Iceland. I have a center there too, home, and then one maniac called and left, he sent a message, email, but voice email. He was disturbed, a young fellow, like 25, 30 years old. He said he just graduated and he's trying to talk to me, find out more. I mean, he's really disturbed. He thought that I'm hiding things and the government is hiding things. It's awful, really. So I'm scared, but what can you do? So now what we try to do is we want to have similar concept like Panacea, but for viruses. Okay, so develop a new concept of bio-nanosensor, right? And it should be very, very reliable for virus detection. Remember, we had the PCR. So in fact, I came here first time some years ago, and I felt totally fine. And in order to enter the TII, they needed the PCR test, and I had COVID, and I didn't feel anything. So the PCR test was always miserable. Yeah, you give that, they give you this thing, and then after 24 hours or 18 hours, 10 hours, they send you this thing. So it's too much big machinery, we called it that time. So can we do that automatically, for example, right? All these byproduct sensors. And the thing is, the viruses hijack the injected cells' molecular machinery to make copies of themselves. That's really the dangerous part of it. Same thing with the cancerous cells, right? These sensors monitor the excessive byproducts of this replication, such as proteases. And then antibody sensors. So these are the two type sensors that we want to develop. Immune system recognizes viruses by their antibodies binding to virus antigens, and antibody tags a virus or an infected cell for attack by other parts of the immune system, or can neutralize its target directly. So that was our project, and there are many details here, right? And that's what we said, that this IoBNT concept can be adopted to, not only COVID-19, but there are many, many, other virus diseases to solve this problem long term, right? So that's the point here. And so, thank you. And if you have questions, I'll be glad to answer to you. It's not easy to talk about many details because we have, interdisciplinary work here. And so I didn't want to go too deep into issues here. But believe me, there are many, many details that is very fascinating. And I started this research, I told you, like 18 years ago. I wish I have 45 years career in communications. So I wish I started this right in the beginning, and it's really fascinating. But anyhow, I still have, I hope, some time to work and continue to contribute. Thank you.